everyone. I hope all of you are doing pretty good. And I'm back here with another session to complete this particular chapter, right? So I really hope that you guys revised what we learned in the previous session. And today we are going to move forward with the further topics. So today we are going to cover the topics movement of substances through membrane. Now I know I did tell you about the movement of substances through membrane in the previous session as well, but today we'll dive into just a little more technical stuff, okay? And then we'll be learning about what exactly is the cytoplasm or nucleus, although I did brief you guys about the different cell organelles, but as I told you, we'll be doing all of these in detail today, right? So we'll be going through the nucleus, ribosomes, endoplasmic reticulum, and Golgi apparatus. Now, the important part for you here is that most of the questions that you'll encounter in the Olympiads or in your subjective question papers of the school, you'll see we'll have the nucleus and from the endoplasmic reticulum. Now, these are the most important organelles of the cells from which the questions are frequently asked. But don't you worry, we'll be covering all of that. Okay. So starting with the movement of substances through the membrane. Now, the most important thing that we studied in the previous session was that movement of substances or the components through membrane is directed because the membrane is selectively permeable. And by selectively permeable, I mean it senses what needs to be allowed to enter and exit through itself in order to maintain the balance in the cell, right? Now we'll be learning about how exactly beside the transport proteins, what are the factors help in the movement of substances across the membrane, right? Now we'll go through this definition, mark all the keywords because those are like really important for you to memorize in memorizing in all of these definitions, okay? So first things first, diffusion. Diffusion. You should understand what is diffusion first of all. Diffusion is the movement of substances from their higher concentration to their lower concentration. Now, the most easiest example that comes into my mind in order to explain this particular terminology is suppose you have a deodorant in your one hand, or let's say you have a perfume in your one hand, okay, and you're standing in the corner of your room, the farthest corner of your room. Now, if you spray a little perfume in that particular corner, what will happen? After about 2 to 3 minutes, the perfume fragrance will be spreading throughout across, like throughout the room, across the entire corners of the particular room that you were standing in, right? So this is the diffusion. How is it diffusion? Because the corner where you were standing, you sprayed the perfume, and that in that particular corner, the perfume was present in the higher concentration. All the droplets were present in the higher concentration, and then they migrated towards the areas which did not have those droplets. So they moved from their higher concentration to their lower concentration, right? I hope this terminology is clear to you. Next thing is when we discuss the diffusion in the context of the membrane, you see, we have the diffusion through permeable membrane, selectively permeable membrane. Never forget to mention the selectively word with the membrane, okay? Moves the substances from an area of high concentration down its concentration gradient. Down its concentration gradient. Concentration gradient means difference in the concentration of two things. Sorry, difference in the concentration of one thing at different places, right? So from higher concentration, it is moving towards its lower concentration. And this is a passive form of transport. Okay, there are two types of transport: active transport, passive transport. Passive because it does not use any energy, it is just moving down the concentration gradient. Okay, it is just moving along according to its concentration in a particular medium or different cells. And active transport uses the ATP, which is the energy, universal energy, and that energy which is used by the active transport makes it active because it can move against the concentration gradient or along the concentration gradient, okay? Then we have the diffusion and osmosis. Diffusion I've already explained to you people. Osmosis, let me explain again. Osmosis is the movement of water particles or solvent particles from their higher concentration to their lower concentration through a semi-permeable membrane. Osmosis is basically identical as diffusion. Only thing that you have to change and mention is, first, it is always about the solvent molecules, okay, the water molecules. And secondly, it is always through a semi-permeable membrane. Diffusion can be happening in the solid, gas, and liquid, but osmosis only occurs through a semi-permeable membrane and only in regards to the solvent molecules or the water molecules. Okay, keep that thing in mind. Now see, and this is how the molecules move across the membrane through the diffusion. Okay, now what you see right here is the water soluble molecules. Water soluble molecules, because they kind of get dissolved with the water and get transported across the membrane pretty nicely. So what you see right here is that this is the channel protein or the internal protein that we talked about, and water molecules will be moving across the plasma membrane in and out of the cell based upon their concentration gradient. Okay, and fat soluble molecules, they do not require any kind of transport protein. Why? Remember when we were talking about how exactly the lipid bilayer will have the lipid molecule, and that lipid molecule will have a hydrophilic head and hydrophobic tail. Okay, hydrophilic head, hydrophobic tail. So what you see right here is that the transport protein is required for the water soluble molecules because it cannot pass through the membrane. Why? Because the membrane from the middle, from in between the membrane, is made up of the hydrophobic molecule or the water fearing molecules, the tail of the lipid molecule. Okay, but the fat soluble molecules, the lipid soluble molecules can pass across the membrane quite easily. Okay, so that is one thing, and that is how the movement of substances takes place throughout the membrane. Next thing that we are going to study is the cytoplasm. Now, the most important thing about the cytoplasm is that cytoplasm determines if a cell is living or non-living. If the cytoplasm of a cell is moving, the cell is living. If the cytoplasm of the cell is not moving, then that particular cell is dead. Okay. Now, important thing is cytoplasm is the thick solution. Why is it thick? Why is cytoplasm thick? Because it contains all the cell organelles. It contains all the molecules of proteins and fats and carbohydrates which are traveling through the cytoplasm and moving and being exchanged from in between the organelles. Okay, that is why it is thick, because it has a lot of materials dissolved in it and moving through it, and it fills each cell. It is enclosed by the cell membrane, right? And it is mainly composed of water, salt, and proteins, just what we discussed now. Okay, and in the eukaryotic cell, remember when I told you about the prokaryotic and eukaryotic cells? In the eukaryotic cells, the cytoplasm includes all the materials inside the cell and outside the nucleus. Okay, now let me tell you exactly the role of the cytoplasm is what we'll be discussing first of all. Okay, so cytoplasm is the site of metabolic reactions. Okay, metabolic are the kind of chemical reactions which occur inside the living organism, so it is very important for you to understand. Okay, so cytoplasm, first of all, it is the site of all the metabolic reactions. Metabolic reactions. Okay, and what are the metabolic reactions? Metabolic reactions are the reactions, this is how you write the reaction in the short form: R, subscript F, X, and superscript N. Okay, so reactions occurring in a living organism, living being. Okay, for example, the reactions which are going into our body right now are also called as the metabolic reactions. So let's say all the reactions which basically are occurring inside the cell, they are occurring inside the cytoplasm, and that is why cytoplasm is the site of metabolic processes or reactions, right? Next thing is that it brings about the exchange of materials between the cell organelles. Didn't I mention this to you before as well? The moving cytoplasm means living cell. Non-moving cytoplasm or sedentary cytoplasm means dead cell. So if the cytoplasm is moving, it is trying to exchange the material between the different cell organelles which are present in the cell, and that is why it helps in the exchange of materials. And the moving cytoplasm is the key feature of the living cell. Okay, so I'm going to write that here. Exchange of materials or components, whatever suits you, between the cell organelles. Cell organelles. Okay. Apart from that, it also is a storehouse for the raw materials which are required for the metabolic reactions. For example, let's say like my cells, they have mitochondria inside themselves. Okay, and my mitochondria requires glucose for the sake of producing of energy. Okay, so the glucose will be just floating around like this in the cytoplasm. Okay, freely, and then cytoplasm will be like bam, mitochondria needs the glucose, it will start moving, it will transport the glucose to the mitochondria, and that mitochondria will produce ATP or the energy with the help of that. Okay, so storehouse for the raw materials. Storehouse for raw materials. Please note this down with me. I hope you are noting this down. Raw materials for metabolic reactions. Metabolic reactions. Okay, now that being said, what you see right here in this particular diagram is that this is the cell. Okay, and this cell is enclosed by the cell membrane, and it contains the cytoplasm. Cytoplasm inside it, right? That is what you need to learn about the cytoplasm. Now we'll be moving forward with the animals and the plant cells. Okay, observe this particular diagram of animal and plant cell. I think I did tell you about how exactly plant and animal cells are different from each other, right? They basically have, if we talk about the plant cells will have a lot of chloroplast, which are called as the kitchen of the cell, right? Because they help in doing the photosynthesis and hence producing the food. Whereas the animal cells will not have any kind of chloroplast. Apart from that, plant cells, they'll always have the cell wall because they cannot move from one place to another. So in order to, be themselves or protect their own structural integrity or maintain the shape of their cells, they have the cell wall. Whereas the animals, they can move from one place to another. If the cell wall was present in the animal cells, will be very rigid and will be moving like this, which I don't think will look nice, right? So that being said, you can observe different kind of differences or different kind of structures which are present in the both animals and the animal and plant cell. Okay, that being said, I'll be providing you with the difference table because that is like extremely important. Okay, and we'll discuss all of these one by one. Right, so starting with the plant cell and the animal cell, plant cells are usually larger. Why are they larger? They need to have a cell wall, they need to have the chloroplast. Okay, and they need to have a lot of things floating into their cytoplasm for the sake of doing all the metabolic reactions. Animal cells are smaller than the plant cells. Also, animal cells basically are present in the locations according to the requirement and according to the functions. That also affects their shape. Okay, and then in the plant cells, cell wall is present, right? And here the cell wall is absent, what we have been discussing from past few slides. Then plasmodesmata is present in the plant cell and it is absent in the animal cell. Now, what is plasmodesmata? Let's say these are the two plant cells, okay, which are situated adjacent to each other like this. Now, they will have to have a cytoplasmic connection or a connection of both their cytoplasms in order to communicate and work in a synchronized manner. Okay, and that particular connection will be called as the plasmodesmata. So what is plasmodesmata? Plasmodesmata is the cytoplasmic connection between the plant cells. Okay, and then chloroplast is present, chloroplast is absent here, which is quite obvious, right? Because we cannot do the photosynthesis. And then one more important difference is the vacuoles in the plant cells are large and permanent, and in the animal cells the vacuoles are small and temporary. How exactly this difference comes into play? For example, let's say there's a plant and that plant requires to store a lot of oil or a lot of rubber or a lot of gum. Okay, as you see, we basically obtain the rubber and gum from the plants. Okay, so what will happen, the plant cell will have to have a large central vacuole in which it will put all the materials which needs to be stored. Okay, and then that will be permanent there. Okay, and in the plants, in the animal cells, the vacuoles are very, very small because at times we just need to store extra water, maybe, right, or extra salts maybe, and whenever those are required, those are used, and then vacuoles just vanish. Okay, moving back to the particular diagram, what you see in this animal cell, can you find the vacuole? Can you find the vacuole? No. But can you find the vacuole here? Yes, the large central purple colored vacuole, right? Now moving forward to the next differences, we have the tonoplast. Okay, let's say if this is the cell and this is the large vacuole that is present, so the membrane of vacuole will be called as the tonoplast. Membrane of vacuole will be called as the tonoplast. Okay, so tonoplast is present around the vacuole and tonoplast is absent because there is no particular vacuole per se. Okay, and then centrioles are absent. Centrioles are absent and centrioles are present. For you to understand what basically are centrioles, will be starting with the cell division. Okay, let's say two cells need to be divided, right? We'll be moving to the nucleus in the next slide. So yay. Right. So let's get back to the topic. Let's say if this cell needs to divide into the two cells. Okay, that is how division takes place, right? One cell give rise to the two cells, two new cells. So let's say we have the chromosomes or the DNA arranged right here in the middle of the cell. What are chromosomes? Chromosomes are the condensed or super folded form of the DNA. Okay, now, centrioles are the small organelles which will be present which will be present near the nucleus inside the cell, and whenever the cell needs to divide, and these centrioles are always present in the pair, so these will move across the opposite poles. Okay, like this. And once they have moved the opposite poles, they will start attaching themselves with the chromosomes like this with the help of the filaments that they secrete. Okay, and after this happens, they start to pull the DNA into half towards the opposite side, and once that is done, two separate nucleus are formed and the DNA is divided into half. Okay, so these are the centrioles. These basically help in cell division. In cell division. Okay, if you do not understand this, don't worry about it. You can ask your teacher or you can ask me. Okay. Next thing is nucleus is present along the periphery in the plant cells and in the center of the animal cell. Why? Because the nucleus goes to the boundary as the central vacuole is present in the plant cell, where the nucleus remains in the center of the animal cell as there is no particular vacuole hogging its place. And then lysosomes in the plant cells are rare and lysosomes are present in the animal cells. What are lysosomes? You must have learned about lysosomes a lot in the lower classes as well. Suicidal, these basically are the suicidal bags. If they burst, they are going to eat their own cell. They are going to digest their own cell and cause the death of the cell. Next thing is storage material is the starch grain. Why starch? Because during the photosynthesis, when the glucose is made, a lot of glucose molecules come together to form the starch molecules, and that starch molecule is stored into the plant cells and so in the form of stored food. Whereas in the animal cells, glycogen granules or the glycogen molecules are the ones which are used as the stored material. Okay, now I know this might be a lot, but it's very easy if you constantly revise it. Okay, moving to the nucleus. Nucleus is like my favorite organelle. Why? Because it is the brain of the cell, and secondly, it is way too easy to learn. Okay, so first of all, you need to understand that nucleus is a membrane bound organelle. By membrane bound organelle, we mean that nucleus is covered with the help of a membrane. Okay, all the nuclear components are present within an enclosed membrane like this. Okay, and it contains the DNA, which is the genetic material. And I did discuss this in the previous session as well. DNA, or the genetic material, is the hereditary material. What is hereditary material? Which is passed on from one generation to the another generation, right? Now, coming back, it is present in the eukaryotic cells. Remember, true karyotic nucleus. Okay. Next thing, major work is it helps in the transcription and replication processes. Let me tell you what is replication. When DNA is made from DNA, that is replication because it is replicating, it is making the copies. What is transcription? When with the help of the DNA, we make the RNA. What is RNA? RNA is ribose nucleic acid, and RNA is mostly present as a genetic material in the viruses. Then why do we need RNA? Because RNA helps in making up of the proteins in the body, and our entire body is made up of what? Proteins. Got my point? So transcription is making RNA from the DNA. Next, largest organelle inside the cell, which takes the entire cell's 1/10 volume, is the nucleus. Now what you see right here is, this is a picture. This purple picture is what you see is the nucleus, and it basically is all haphazard manner because it denotes how the chromatin material or the DNA or the genetic material is basically lying around in the nucleus in the form of a thread. Whereas in this particular structure, what you see, this particular part, the purple colored structure, this is what we have as the nucleus. Okay, and surrounding it is the endoplasmic reticulum. Endoplasmic reticulum, right? And we have several other organelles as well. So we'll do one thing. I'll draw a stick diagram of nucleus for you, and that will be very easy for you to learn. Okay. So when we draw the nucleus, nucleus is a membrane bound organelle, and it is a double membrane bound organelle. That means it has two membranes. Okay. It has two membranes. So this is the nucleus that I'm drawing for you people, and that is the nucleolus, and this is what we call is the DNA material, and it will have small proteins on itself which will help in the proper what do we call it, which will help in the proper folding. And then this is the nucleoplasm. Right? Now listen to me very carefully. As I said, nucleus is a membrane bound organelle, but it's a double membrane bound organelle. This is the outer membrane of the nucleus, outer membrane. All right. And this is the inner membrane of the nucleus. Now you must be thinking, ma'am, why haven't you drawn the membranes continuously? Why are there like small breaks in between of them? Because these breaks, they kind of denote the pores. They are the nuclear pores. Nuclear pores. It is very important for you to understand that nucleus is not a continuous organelle. It's not a organelle with the continuous membrane. It will have small pores inside of it. Why is there the requirement of pores? Because those pores are very important for the sake of communication between the nucleus and the entire cell. In order to pass on the instructions to the entire cell, nucleus requires the pores through which all the instructions will float out in the form of the mRNA, and then those mRNA will be used for the sake of making the proteins in the cytoplasm. All right. Now that being said, this is called as the nucleoplasm. Nucleoplasm. Right. Remember when I told you about what is a cytoplasm? Nucleoplasm is the same thing. It is just as present in the nucleus. Okay. And then we have the DNA material. DNA. Listen, there are small proteins which are called as the histone proteins, which are present in the DNA, and DNA lies in the unfolded state in the nucleus. Okay, these small proteins, they help in the superfolding of DNA and making the chromosomes that we just studied about. Okay, they help in the superfolding of the DNA. Now, one thing that we are left with is the nucleolus. Nucleolus. Okay, what is the function of the nucleolus again? Nucleolus helps in producing or making ribosomes, which is the another cell organelle of the cell. Okay, now listen to me. Ribosomes, they basically help in making proteins. They help in making of proteins. Okay, now that being said, this is the entire nucleus that you need to revise, and this is the brain of the cell. It controls everything inside the cell. Okay, every instruction is just basically controlled by the nucleus. Next thing that we are going to study about the ribosomes. Now, I just told you who makes the ribosomes. Nucleolus. What is the function of the ribosomes? Making of proteins. Okay, see, ribosomes basically are the minute particles, okay, and they have a lot of RNA, okay, and proteins. How do they have the RNA and proteins? Because that there's a particular process which is called as the transcription that we discussed. Transcription is RNA being made from the DNA, and then after transcription we have the following process which is called as the translation. In the translation, mRNA which was produced by the nucleus will move out from nucleus to the cytoplasm, where ribosomes will come and attach itself to it, and then they'll help in making the proteins with the help of the instructions which are encoded in the mRNA. Okay, if you don't understand this, that's completely fine. That is not what you need to learn for the 8th standard, but it will be surely appreciated if you try to understand it. On top of that, we basically have all of this being repeated in the 9th standard as well. Okay, right. So we have the function of ribosomes is to synthesize proteins, which we just talked about. Okay. Now, see, apart from that, proteins basically make up the entire body. They help in making up your body. They help in repairing the damages of your body. They help in doing all the metabolic reactions which are present in the body. Okay. And these ribosomes, on the other hand, they could be freely floating in the entire cytoplasm after they are made into the nucleus. Okay. Now, one thing that will be moving forward with the ribosomes is, you see, ribosomes, this is the endoplasmic reticulum. Endoplasmic reticulum. Okay. If we talk about the endoplasmic reticulum next, endoplasmic reticulum is the organelle which is present in continuation with the nucleus. Okay, so it's like the membrane which is making the nucleus is continuing forward and making the endoplasmic reticulum as well. That endoplasmic reticulum is found in just the eukaryotic cells and not the prokaryotic cells. Okay, and it has multiple functions which we'll be talking about. Major thing, endoplasmic reticulum, first of all we'll discuss about the types and then we'll discuss about the functions separately. Okay, so we have the SER and we have the RER, smooth endoplasmic reticulum, okay, and rough endoplasmic reticulum. Now, what you see right here is that rough endoplasmic reticulum is present just next to the nucleus, whereas the smooth endoplasmic reticulum is present in continuation with the rough endoplasmic reticulum. So it's like first there is nucleus, then there is rough endoplasmic reticulum, then there is smooth endoplasmic reticulum. Okay. The thing is, Bacha, why is it called as the rough endoplasmic reticulum and why is it called as the smooth endoplasmic reticulum? Because in the rough endoplasmic reticulum, we have ribosomes embedded inside it as small beads, which makes the texture of this particular endoplasmic reticulum a little rough. Whereas in the smooth endoplasmic reticulums, there is no ribosomes at all. Okay. And since there are no ribosomes at all, the structure or the texture of that particular smooth endoplasmic reticulum is smooth as the name suggests. Okay. That being said, you should understand that this rough endoplasmic reticulum is present right next to the nucleus, and the smooth endoplasmic reticulum is present in continuation with the rough endoplasmic reticulum. Okay. Now let's discuss about their structure, what exactly these are made up of. The rough endoplasmic reticulum, it could be made up of ribosomes and it has a lot of tubules and membranes, whereas the smooth endoplasmic reticulum, it just made up of the vesicles. Vesicles are the small bubble-like structures which are present in the cell, and it is made up of lot of tubules. Okay. On top of that, the SER and RER, the smooth endoplasmic reticulum and rough endoplasmic reticulum, they differ a lot in terms of the functions as well. Okay, so we'll be talking about their functions next. Now, this is going to help you in understanding the function, the structure, and it is also going to serve as a difference table for you. Now, what do you see? SCR does not have the ribosomes. RER possesses the ribosome. On top of that, major function of the SCR is the synthesis of lipid. Since the SCR or the smooth endoplasmic is doing the synthesis of lipids, it also is known to done the biogenesis of membrane. Biogenesis of membranes. Now, these membranes could be the membranes of cell organelles or the cell membrane or plasma membrane, however you want to call it. Okay. And biogenesis means making up of membrane. Okay. Making up of something or producing something, right? Producing something naturally. So you see, remember when I told you that membrane is made up of the lipid bilayer. There are two layers of the lipid. So SCR is what it is doing. It is synthesizing the lipids. And if it is synthesizing and producing the lipid, it is helping in the biogenesis or making up of the membrane. And then the RER, since it has the ribosomes, it is going to produce the proteins, right? On top of that, the SCR is formed of vesicles and tubules. As I have just told you about, tubules are the tube-like structure, vesicles are the small bubbles which are found in the cytoplasm, whereas the RER is formed of the tubules and cisternae and ribosomes. What are cisternae? Cisternae are structures like this, which are present in continuation like this. Okay, like this. Coming back to the diagram, you can see cisternae right here. Okay. These are the cisternae. Now back to the table. The smooth endoplasmic reticulum, it is found in the periphery. Periphery means it is found on the boundary. And the rough endoplasmic reticulum is found in the cytoplasm deep or in the center. Why? Because let's say if this is the cell that we are talking about and this is the nucleus which is present in the cell, the smooth endoplasmic reticulum and rough endoplasmic reticulum, the rough endoplasmic reticulum is present like this. Okay, this is the rough endoplasmic reticulum. And the smooth endoplasmic reticulum will be present like this, right? Because it is present in the continuation of the rough endoplasmic reticulum. So this is present in the periphery or in the boundaries, okay? Whereas the rough endoplasmic reticulum is present in the center or deep inside the cell. Apart from that, the rough endoplasmic reticulum may give rise to the smooth endoplasmic reticulum, and the rough endoplasmic reticulum could be made up of the nuclear envelope. Okay, now these are the differences which you really need to revise and learn. Okay, and now we'll be moving forward. Moving forward to the next organelle, which is called as the Golgi apparatus. Important part about the Golgi apparatus, I think I did tell you about the Golgi apparatus literally yesterday as well. Golgi apparatus is basically the, let's say, postman of the cell. Okay. Now, when I say postman of the cell, the major function of the Golgi apparatus is taking up of all the proteins and the molecules which are synthesized by the endoplasmic reticulum in the form of the vesicles. Okay, those will move towards the Golgi apparatus. Golgi apparatus will then read, oh, okay, this protein belongs to this organelle, this protein belongs to this organelle, and then that organelle will be like that particular protein will be dispatched to the destined organelle from the Golgi apparatus itself in the vesicles. Okay, and they have a particular structure as well, which we'll be discussing about. So Golgi apparatus, first of all, is called as the Golgi complex or the Golgi body, and it was discovered. Okay, one thing you should know, that whenever we are talking about the organelles, we do not say that they're invented. We say that they were discovered because they were already present there. Okay, so it was discovered by Camillo Golgi, and hence the name is the Golgi apparatus, and this occurred in 1898. Right? That being said, it is located in the cytoplasm, right, next to the endoplasmic reticulum. Why? Because all of the stuff from the endoplasmic reticulum needs to go directly to the Golgi apparatus first, and then Golgi apparatus will dispatch the proteins towards all the cell organelles in the cell. Okay. So let's say endoplasmic reticulum is a factory, and then that factory will give all of its product to the Golgi apparatus, which is the distributor or the postman. And then that postman or the distributor will distribute all the proteins according to the requirement and the destinations of, like, destinations being the target organelles, right? Now see, the structure of the Golgi apparatus is very, very peculiar. It's very, very particular, which we'll be talking about first. Okay. So Golgi apparatus has like two phases, and there we go. Right. So what do you see in the Golgi apparatus right now is that it has two phases. When I two phase, it's a cis phase, and then it is a trans phase. Okay. Cis phase is the receiving phase. Receiving phase. Okay. Everything that is made into the endoplasmic reticulum will dispatch from it in the vesicles. Okay, so the bubbles will have the proteins, and those vesicles will come towards the cis phase of the Golgi apparatus. And when they come towards the cis phase of the Golgi apparatus, they merge into the membrane of Golgi apparatus. So let's say, look at my video. Let's say this is the vesicle. Okay, this was dispatched from the endoplasmic reticulum, and this is the Golgi apparatus. Now this will come towards the Golgi apparatus, and it will merge into the membrane of the Golgi apparatus like this, and the molecule which was being carried by the vesicle will be released into the Golgi apparatus's lumen, which is right here. Okay, this lumen or the tunnel like structure is the one thing where the component from the vesicle was released. Okay, now you can see that this is also made up of the cisternae and the vesicles and the tubules. Okay, that being said, after this particular vesicle is received, it is then processed through the multiple lumens of the Golgi apparatus, and it is labeled with the help of another let's say protein, or labeled just for the sake of dispatching it towards the correct organelle, okay, or the target organelle. Now what really happens in the trans phase? This is the dispatching phase. Dispatching phase. What will happen here? You see, vesicles are forming, right? Vesicles are forming, right? So all the proteins will be, let's say, look at my video again. So this is the component which needs to go out. So it will go and push towards the trans phase of the Golgi apparatus, and trans phase of Golgi apparatus will be formed into a vesicle, and it will dispatch, or it will pinch off from the Golgi apparatus's membrane. Okay, and these are also called as the secretory vesicles. Okay, or the transport vesicles. Transport vesicles. Okay, and the vesicles which are like coming towards the cis phase are called as the incoming transport vesicles. Okay, so that being said, after the like from the Golgi apparatus, time for the questions. Okay, question that I have for you is function of nucleolus. Next question that I have for you is who is postman of cell? Postman of cell. And another question that I have for you is who or what helps in membrane biogenesis? Membrane biogenesis. All right. Now see, what is the function of nucleus? Produces ribosomes. Okay. What is like who is the postman of the cell? Golgi apparatus. Just talked about it. Okay. And who helps in the membrane biogenesis? Come on, this is the easiest. Smooth endoplasmic reticulum. Okay. That is all for today. Thank you so much. You guys stay safe and enjoy. And I really hope to see you in the continuation in the next session. Learn a lot. And that is all for today. Take care.